Okay, great. So I think let's get started. It's my great honor to introduce today's speaker, Dr. Cai Shang from Westlake University. Shang did his undergraduate in biology at Peking University, which is followed by a PhD at Indiana University in the United States. He received an excelled training in cell biology under the mentorship of Claire Wozak. And his PhD work reviewed a comprehensive mechanism explaining how microtubule dynamics supports mitosis. Because I was in the same field with Shang at that time, I read many Shang's inspiring papers and was indeed impressed by the numerous beautiful images and movies. After finishing his PhD, Shang moved to the Silicon Valley and did a postdoc with Mike Clark in Stanford. There, he worked on stem cell in mammary gland. He identified a new population of stem cell that is required for maintaining mammary gland homeostasis. He also identified a stromal niche signaling for epithelial stem cell in mammary gland. Starting in 2017, Shang established his own lab at Westlake University. His research focused on the role of stem cell in the morphological and functional integrity of mammary gland during both physiological and pathological conditions. Excitingly, I think early this year, Shang's lab published, I think, a groundbreaking paper on Cell, showing that the bacteria inside breast cancer cells promote metastasis. And during metastatic colonization, these intratumor bacteria can promote host cell survival by reorganizing actin cytoskeleton to enhance host cells' resistance to fluid shear stress. In this work, I think Shang beautifully combined his PhD expertise in cytoskeleton cell biology and the postdoc experience in mammary gland and create a completely new research paradigm. As you probably heard before the seminar starts, so I chatted with Shang about the discovery. I think it's a really exciting story on how making new discoveries in science. And so I think this is such an influential work, which opened, I think, a really exciting interface between microbiology, cell biology, and tumor biology. And I could not wait anymore to learn the details. Shang, the floor is yours.
Okay. Well, thanks, Xiaolei, for the kind introduction. And also thanks for Chuan for the kind invitation. It's my great pleasure to have this opportunity to share a little bit of our recent progress on the physiological role of the intratumor microbiota in the tumor progression process. And the title, the story will be the lurking culprit of cancer in the background. So first a little bit about the lab interest. Our lab is actually very interested in the breast tissue and the breast cancer field. And we know that the breast tissue is actually constituted by a hierarchy of cells, with the top hierarchy is the stem cell. And these stem cells are long-term survival cells that they can be transformed into tumor-initiating cells. And these tumor-initiating cells can, first further develop and prime the tumor primordia. And these tumor cells later, recruit other normal cells and form a complex tumor ecosystem, including various cellular components. And later, the tumor cells can, progress to invade or intravasate into the blood vessels and travel to the distal organ and initiate a new cancer. In this process, we are actually very interested in, what are the driving forces for the tumor transformation and also the tumor progression, especially at the very early stage of the tumor that we are largely unknown for these early stages. So specific for the cancer progression, we know that cancer cells cannot survive by themselves. They are actually, their survival is actually facilitated by a variety of components, including the cancer-associated fibroblasts, endothelial cells, immune cells, and in recent years, we found that there are some nerves innervation in the tumor tissues. And all these cellular components actually coordinate with each other and support the cancer cell survival and trigger the cancer cells to progress to the malignant stage. So theoretically, if we understand the gene expression and the genetic information of all these cellular components, we should be able to, predict the cancer progression precisely, and how these cancer cells, they gain the ability to progress to the next malignant stage. And indeed, if we use the gene expression profile and use, for example, in the breast cancer field, if you use a PAM50 index, and you can actually categorize the cancer patient into different groups, including the luminal A, luminal B, HER2, basal, and Claudin low and normal. And these different tumor types actually is associated with very distinct prognosis patterns. Some of them have very good prognosis, for example, luminal A and luminal B, and some of them, they have very bad prognosis, for example, the basal-like and also the Claudin low. So, but if you plot the survival time for each specific tumor type, for example, luminal A, luminal B, HER2 basal, you find that these patients, they actually have very different survival time, big variation from several years to like decades. And this indicates that besides the genetic information and transcriptome information of these cellular components, there's maybe some other factors involved in this process that we are unknown. And identifying these new factors involved in the cancer progression is probably will help us for the next step of precision medicine. And what the question is, what other unknown components could be involved in this system? And the experiment 100 years ago probably gave us some insights. This is the famous Coley's Toxin assay. And Dr. William Coley actually used two strains of bacteria. One is Streptococcus pyogenes, the other is Serratia marcescens. And he injected these two bacteria back into the patient tumors and triggered the immune response that can efficiently shrink the tumor and improve the prognosis. And for some of the patients, they can the tumor will shrink and ultimately disappear. But the question is, why is the bacteria that triggered this experiment to trigger this immune response specifically towards cancer, against the cancer cells? And why bacteria trigger specific cancer immunity? And this question has now been solved and answered. But actually, if you read the paper from William Coley 100 years ago, he actually gave us some answers. And he said that the explanation is not so difficult. He believes that these all variety of malignant tumors are actually of extrinsic and microbic origin. So he thought that these tumors probably during the initiation and the development, the progression, there might be microbial components involved in this process, but he didn't show any evidence of that. And after that, for many years, there were no or rarely any evidence showing that cancer cells contain microbic components, because we generally believe that our immune system can clear all the microbes and keep it in a sterile environment. But due to the development of the next generation sequencing, we can see actually a very trivial amount of the DNA information from the samples. And there are multiple papers reporting that besides the GI tract tumors, it's not surprising that GI tract tumors, they contain some bacteria because in the colon, there are a huge amount of bacteria there. But there are also some papers reporting that in the non-GI tract tumors, for example, the breast cancer, lung cancer, pancreatic cancer, they also reported detection of the bacteria signals from these tissues. But the question is, these low abundance of the bacterial information, are they the true bacterial components within the tissue, or they are actually the contamination from the environment? This is a serious problem in the field. And this problem has been solved by two beautiful papers published on Nature and Science in the year 2020. In the year 2020, Rob Knight lab that actually published a Nature paper. They assembled a cohort of the samples from the TCGA, around 20,000 samples, the sequencing data. They applied a very stringent decontamination pipeline. After removing these contamination signals of the bacteria, they still can see tumor type associated bacterial signatures, suggesting that these but the data target the different region of the 16S ribosomal DNA, and to minimize the contamination. And they characterized more than 500 patient samples, tumor tissues, and they found that the presence of the bacteria in this breast tissue, in the cancer tissue, is actually comprehensively or broadly exist in various type of breast cancer, various types of cancers, including the breast cancer, bone cancer, pancreatic cancer, GBM, ovarian cancer, lung cancer, and melanoma. And interestingly, these bacterial information actually not the highest in the tissue that more likely to expose in the environment. For example, the lung cancer and melanoma, they actually remain relatively low. But the bone cancer and the breast cancer, you see that the bacterial signal is quite high, suggesting intrinsic problem. Also, interestingly, present and broad progression. System tissue, whether it's these bacterial information, try in the tissue even in the water, increase the sensitivity and accuracy. So we did two things. One is to use the base shearing to try to release as much as the bacterial information from the tissue, because some of the bacteria, they have very thick cell wall. We need to release them as much as possible. And the second is we control the contamination from the reagent. We try to minimize the bacterial contamination signal in the reagent and get the sensitive detection. So later, after we optimized the system, we found that our NTC, the non-template controls, actually can allow us to detect the 10 to the second bacteria in our sample. And this sensitivity will allow us to see whether there's a low abundance of the bacteria within the tumor tissues. So we next tested in the mouse, the famous mouse breast cancer model, which is MMTV-PYMT. Most of the almost 100% of the mouse, they will develop breast tumor, and most of them will develop lung cancer, lung metastasis, sorry. So with our optimized protocol, we found that even in the normal breast of the MMT-PYM in the mouse, we can detect a very low amount of the bacterial information, the genetic information in it. And if you test the breast tumor, you can see that this abundance will be increased by tenfold. But this is just the genetic information. We also tried to culture these tissues to see whether they are live bacteria within the tissue, and we found that these bacteria are indeed they are live organisms in the tissue. They can grow in the culture plate. So the next question is, if they are there in the breast tumor, where do they localize, and whether they are uniformly distributed in the tissue or they have specific localizations. So we stained the tumor tissues with epithelial keratins and also the bacterial informations. And also we stained the immune cells by the macrophage by F480. We found that after quantification, we found that most of the bacteria within this tissue is actually associated with epithelial cells, about 80%. But a small amount of the bacteria is associated with immune cells, the CD45 positive cells, and some of them are within endothelial cells. But interesting thing is that we found that F480 macrophages, they are present in the tumor, but they do not colocalize with the bacteria very well. And this brings up an interesting question that if the immune system is in the breast tissue, breast cancer tissue, why they did not clear the bacteria within the tissue. One possibility is that this immune system is impaired. They are functionally not proper for clearing the bacteria, and there might be other possibilities. So this question has been solved after we did the EM's images and to try to find and pinpoint those microbial structures. And after the EM analysis, we found that most of the structures we identified, like bacterial-like structures, they're actually reside in the cytosol. About 90% of the more than 90% of these structures actually in the cytosol. Between the cell in the intercellular space, we rarely see these structures. And after quantification of more than 7,000 cells, we found that about 3% of these cells, they contain these bacterial structures, and this is relatively low abundance. But this nicely explains why these bacteria, they were not cleared by the immune system. They probably enter invade the cells. They hide themselves by the cancer cells and avoid being attacked by the immune system. But if these bacteria goes into the cytosol, they may trigger some innate defense system, and the cell may respond to it and try to kill these bacteria. So the structure we identified in the cytosol, are they live bacteria? Can they survive in the cytosol or not? So we did an interesting experiment. We dissociated the tumor tissues into cells, culture these cells in the culture dish. And we use two types of antibiotics, try to kill the extracellular or intracellular bacteria. And the ampicillin and gentamicin, they can only kill the extracellular bacteria. They cannot go impermeable to the cancer cells. But doxycycline, we know that these antibiotics, they can go and penetrate the cancer cells and kill the intracellular bacteria. So we culture these tissue cells, and we found that these antibiotics can efficiently kill the bacteria in the medium. But if you only kill the extracellular bacteria, the tumor cells can still give rise to these bacterial colonies. But if you kill the intracellular bacteria with the doxycycline, and both inside and outside of cells is clear. And this suggests that if you give the system a environmental stress. I mean, these bacteria, maybe they can go into the cells and survive there and avoid being attacked by the immune system. So these data suggest that in the mouse tumor tissue, it's also not a sterile environment. There are low abundance of the bacteria residing there. And the question is, what kind of bacteria are there? Are there specific types of bacteria within this MMTV PMT tumors? So we try to profile these bacteria, but when we try to profile this bacteria, we face a serious challenge because we know that the bacteria genome size is actually 1 to 1,000 to a cell. And if the bacteria to cell ratio is 1 to 100, this genetic information, the ratio, it will be 1 to 100,000. So to try to detect this low amount of genetic information in the sample is quite challenging. We'll have to optimize the protocol and be able to identify and accurately detect this low amount of the genetic information. So what we did is that we we added a further step with the biotin enrichment step. We used the biotin labeled primer, try to amplify this bacterial information from the sample, and then enrich it and do a second round of the amplification. With this optimized protocol, we were able to detect a 10 to the 4th or 10 to the 3rd bacteria within our tissue using this this is the standard bacterial strain, the E. coli. And this sensitivity actually allow us to detect the bacterial profile within the tissue. So with this optimized method, we quantified the control, which is the environmental bacteria, and the normal breast and the breast tumor. We found that normal breast and breast tumor are contain mainly firmicutes within the tissue, which is strikingly different from the environmental control. The environment control mostly the proteobacteria there. This and the clustering showing that they are quite distinct from the environmental bacterial information, suggesting that these tissues, the bacteria in the tissues, they're not likely to be the contamination. And another interesting phenotype we saw is that the breast tumor contain the diversity. The diversity of the bacterial microbial community in the breast tumor is actually much lower than the control, the normal breast, suggesting that there were less species showing up in the breast tumor, and indicating that the breast tumor is a unique environment that actually enrich specific type of bacteria within this tissue. And what type of bacteria are there? If we check at the genus level, we found that the normal breast and the breast tumor is actually accumulated for 4 major genus. One is the Staphylococcus, Streptococcus, Enterococcus, and the Lactobacillus. So these 4 are the major genus in the breast tumor, and it's different from the normal breast. But interestingly, these bacteria actually we probed in our sequencing data is actually quite consistent with our culture data, showing that the most abundant bacteria we isolated from the breast tissue is actually Staphylococcus, Lactobacillus, Enterococcus, and Streptococcus. And this suggests that our sequencing profiling is actually quite accurate and sensitive. So these data suggest that in the breast tissue, besides those cellular components, endothelial cells, fibroblasts, and immune cells, we do have a new component of the prokaryotic cells, the bacteria. And these bacteria reside in the tissue with the low abundance. And the question is, what are they doing there? So intuitively, these bacteria are supposed not to play big roles in the tumor progression because their amount is quite low. It's hard to believe this low amount of the bacteria play any significant role in the cancer progression. For example, the bacterial cell volume is 1 to 1,000, and the genome also is 1 to 1,000. So how could such a tiny organism trigger a significant phenotype in the cancer progression? But without experiment testing, we cannot draw this conclusion. So we try to develop methods to try to interrogate whether these bacteria are functional or not. If you want to know they're functional or not, you have to eliminate these bacterial components. But eliminating bacterial components using antibiotics will have some serious issues, because we know that all over our body there are quite some bacteria residing in our body, in the colon, in the on the skin, and also in our oral cavity, etc. So if you use antibiotics, you cannot avoid, disturbing these microbiota within our body. And how do you know that the effect you see is specifically triggered by the intratumor bacteria? And we know, and there are many literatures reporting that the gut microbiota actually play important roles in cancer progression. So we try to optimize or develop new methods, try to specifically target the tissue resident microbiota and try to specifically eliminate this population of bacteria. And my postdoc actually did find such a way to do that. And his way is that actually use a combination of antibiotics and using different administration routes, try to clear the bacteria in the mouse. And the first route is through the drinking water, and using this combination of antibiotics through the drinking water. This is quite strong antibiotics. It can efficiently eliminate the gut microbiome by 10,000-fold, and also efficiently eliminate tumor microbiome by 100-fold. But if you administer these antibiotics through the vein injection, intravenous injection, and these antibiotics first arrive in the tissue, and they clear the tissue bacteria, and they play minimal roles in the gut microbiome abundance. You can see that the gut microbiome is actually remain similar level to the control, but the tumor microbiome is actually efficiently reduced. And the third way is using the doxycycline through the drinking water. Doxycycline is a single antibiotics, and when mouse drink the doxycycline water in the gut, the doxycycline does not play significant role in the gut microbiota in the abundance, because this is a single antibiotics, and in the gut there contains a huge amount of microbiota. But doxycycline can be well absorbed by the mouse, and they can efficiently clear the tissue microbiota and reduce it by 100-fold. So with this distinct antibiotics treatment, we can address, specifically what's the role of the intratumor microbiota play in the tumor progression. So after we use these different antibiotic treatment, we found that if you clear the gut microbiome, and the intratumor microbiome, and it efficiently reduce the tumor size, you can significantly reduce the tumor size. This is consistent with the literature that gut microbiome play a role in the tumor growth. But if you only clear the intratumor microbiota, and the tumor growth remains exactly the same, it doesn't play any role in the tumor growth, in the primary tumor growth. And this makes sense because the abundance or the amount of the bacteria in the primary tumor is actually very low. It's hard to believe that they play a striking role in the tumor growth in the primary tumor with this low abundance. But when we check the lung metastasis, and we saw a striking difference between the control and the antibiotics treatment, and if you no matter you clear the gut microbiome and the intratumor microbiome, or you only clear the intratumor microbiome, and you can efficiently reduce the lung metastasis numbers. And this suggests that the intratumor microbiota likely to play a role in the tumor metastasis process instead of the tumor growth. But still has the problem that when we use the antibiotics, you actually also disrupt the gut microbiome. Although the abundance remains similar, but there could be a dysbiosis in the microbiota components, the composition. So to test whether the gut microbiome play a role in this process, so we use the germ-free mice. We actually injected the tumor bacteria containing tumor cells into this germ-free mice. and use the antibiotics to kill these bacteria. And in this way, we only kill the tumor-resident bacteria instead of influencing the gut microbiome, because there's no gut microbiome in these mice. Yeah. And with this tumor model, we see that in the germ-free mouse, the primary tumor growth still remains the same, but the lung metastasis has been drastically reduced by the antibiotics treatment, and this is a several-fold reduction from for the lung metastasis. And this experiment demonstrates that without the gut microbiome, the intratumor microbiota also plays a significant role in the cancer metastasis. So these experiments demonstrate that these bacteria are actually functional within the tumor. Within the tumor tissues, they have important physiological role in tumor progression. But the question is, well, in what way these bacteria influence the primary tumor cells? And since these bacteria, they have very unique localization within the tumor cytosol, tumor cell, the cytosol. And we think there might be two possibilities. One possibility is that when the bacteria invade the tumor cells and the host cells, the host cancer cells, they are activated and acquire a new ability to metastasis. They have better ability to metastasis to the distal organ. And the other possibility is that when the host cell is invaded by the bacteria, the host cell actually releasing some alarming signals to the surrounding cancer cells, and the surrounding cancer cells may be mobilized and they can metastasis to the distal organ better. So to distinguish these two possibilities, we try to isolate the circulating tumor cells and to try to see whether the circulating cells, they carry the bacteria or they don't carry the bacteria. So we actually analyzed quite some mice for the circulating tumor cells, and also co-stain with the bacterial probes. And we found that in the circulating system, the circulating tumor cells, they do contain the bacteria information, the genetic information in the circulating tumor cells. And we observed the two types of circulating tumor cells. One is the single circulating tumor cells, the other is the clusters of the circulating tumor cells. And the previous literature has demonstrated that the clusters of the circulating tumor clusters, they have much better ability to metastasis or survive in the distal organ. And we found that about 50% of these clusters, they contain bacterial information. But this is a snapshot of the metastasis. This is in the circulation system. So we really want to prove, whether the bacteria can travel with the cancer cells together to the distal organ. So we did a genetic lineage tracing assay. We genetically labeled the bacterial strain Staphylococcus xylosis. We isolated from the tumor, and with this a erythromycin resistant cassette and a GFP cassette. And with this genetically labeled strain, we can trace these bacteria, the migration and the dynamics of these bacteria within the tissue. And if we injected this recombinant bacterial strain into the primary tumor, and we found that if you can see the lung metastasis, and this lung tissue will contain the recombinant strain. But if you don't see the metastasis in the lung, the lung we cannot detect this recombinant bacteria. This suggests that the migration of the bacteria to the lung is actually associated with the metastasis. But there's another possibility that the bacteria, maybe they can travel by themselves to the lung. So we injected the bacteria itself into the bloodstream. Or the bacteria invaded tumor cells into the bloodstream. We found that bacteria, when you inject it directly into the bloodstream, they will be cleared immediately, and they cannot colonize in the lung. But if you inject the bacteria invaded tumor cells into the tail vein, and you can detect the bacteria in the lung. This suggests that when the bacteria invaded into the tumor cells, and tumor cell can be hijacked and then travel together to the lung and survive there and establish a new environment. And what kind of bacteria can travel with the cancer cells together to the lung? And we analyzed our microbiome data from the normal breast tumor, the small metastasis, and the macrometastasis to show this progression process. And these bacteria actually clustered into different patterns. One of the pattern showing here is like a sporadic pattern. These bacteria showing sporadically into different tissues, and it didn't show any pattern. We think these might be contaminations or some sporadic bacterial strains. And another cluster is these bacteria, they actually showing up very high in the normal breast, but they get low in the breast tumor and metastasis. And these may be the normal tissue resident bacteria. And there are some other low abundance bacteria. They remain constantly in different tumor tissues. But the major cluster is an important cluster is actually this cluster, showing that initially in the breast tissue it remains low, but when in the breast tumor it increased very high. It maintained this high level in the small metastasis, but when the metastasis growing up in the macrometastasis, it dropped a little bit. And these bacteria, we think that they most likely can travel with the cancer cells together and to the distal organ. And interestingly, these bacteria are actually Streptococcus, Lactobacillus, Staphylococcus, and Enterococcus. And these bacteria are just the strains that we isolated from our tissues. So, and these data suggest that the bacteria can not only affect the metastasis, they actually can travel with the cancer cells together. They can invade into the cells and travel together to the distal organ. And we know that the bacteria or the microbiota we isolated is not a single strain in the tumor. It's actually a community with as a microbiota containing different strains of bacteria. So the question is the microbiota altogether, they cooperate with each other to exert their functions, or each specific strains, they have their role, distinct roles in triggering this cancer metastasis. So we really want to test, the strains we isolated, whether they all have the ability to trigger metastasis or they have distinct functions. So we established this in vitro co-culture system to try to introduce the bacteria into the cytosol and use this as a source to test, for each specific strain, whether they have the function in metastasis. And we found that different strains, they have quite different ability to invade the tumor cells in this organoid culture system. The Staphylococcus xylosis, they have the strongest or the highest ability to invade the cells, and the Lactobacillus. But the Enterococcus is actually the lowest ability to invade into the tumor cells, suggesting that they have distinct they may have different mechanisms invading the tumor cells. But with these organoid containing the bacteria, we try to do functional analysis to see whether these bacteria help the tumor cells to colonize in the lung. So we injected these organoid containing the bacteria back into through the tail vein back into the mouse and let them colonize in the lung, and using tissue clearing imaging to try to quantify, whether the bacteria help the cells to colonize there. And we found that interestingly, the ability for the bacteria to promote the colonization of these organoids is associated with their ability to invade the tumor cells. What you can see is that the strains that invade the cells better, they actually promote the lung colonization better. And the Enterococcus, again, they invade less into the tumor cells, so they have a minimal ability to improve the lung colonization. But this is a part of the metastasis process, and we want to see, for the whole metastasis process, whether the bacteria play similar roles. So we designed this experiment to try to eliminate the intratumor bacteria first, and then reintroduce each specific strains back into the primary tumor, and to try to see these bacteria when they introduce at the physiological level to the tumor tissue, whether this physiological level of the bacteria can trigger the whole metastasis process. And what you can see is that in this MMTV-PMT model, if you use a doxycycline to eliminate the intratumor bacteria, the metastasis will be reduced. But if you reintroduce different strains of bacteria into the primary tumor, you can see the Staphylococcus and Lactobacillus, they strongly increase or promote or recover the metastasis ability, while the Enterococcus introducing into the system, actually they do not play the same role in the metastasis. And this suggests that the bacteria-mediated bacteria, they have a distinct function in promoting the cancer metastasis. And also, since this Enterococcus actually is a very good negative control for the, it's not the bacteria effect, it's the strain-specific effect. It's not likely to be the immune system that triggered by the bacteria and that trigger these effect. But MMTV tumor is actually a tumor with high ability to metastasis to the distal organ. It's an aggressive and a malignant tumor type. We are asking if whether the presence of these bacteria in the non-metastatic tumor types, and they don't metastasis, if you reintroduce these bacteria into the into the cancer tissue, whether they can endow the tumor tissues with the ability to metastasize to the distal site. So we use the MMTV wind tumor model, and this wind tumor is widely known that they don't metastasize very well. And we did the same experiment, clear the intratumor bacteria and re-injected each specific strain back into the primary tumor with only one shot. And we see a striking effect that if you introduce these bacteria we isolated from the tumor back into the wind tumor, we can see that this tumor type, initially they don't metastasize. If you reintroduce these bacteria back into the tumor type, they start to aggressively metastasize to the lung tissue. But again, the Enterococcus didn't play a very strong role in this promoting the cancer metastasis. And this is quite interesting. We think this is quite interesting because clinically, we usually focus on the genetic information or the gene expression information for the patient tissues. And we rarely care about these intratumor microorganism informations. And this data suggests to us that the prognosis of the tumor tissue is not only determined by its genetic or transcriptomic information, but also determined by these microbial factors that we ignored in the clinics. And they may play a role. They may convert a non-metastatic tumor to a metastatic tumor type. So these, to summarize, these data suggesting that these bacteria, the distinct strains of bacteria, they have different roles in promoting the cancer cell metastasis. And we know that these cancer cells, when they intravasate into the blood vessel and extravasate to the distal organ, and it's actually a very harsh process. And the study is showing that more than 99% of the cancer cells, they actually die during this metastatic process due to different stresses in this process. And the question is how the invasion of the bacteria into the cancer cells help these cancer cells to resist to the different stresses in this process and help the tumor cells to survive in the breast in the distal organ. And we know that when we talk about metastasis, we really most of the study actually focusing on these factors. So for example, the EMT, epithelial mesenchymal transition, which promote the tumor motility and also promote the stemness. And this is the widely studied in the tumor metastasis process. And another factor is actually immune system. The immune system also play a key role in modulating the cancer cells' ability to survive or colonize in the distal organ. So how does the bacteria play the role? Does it exert their role through modulating the EMT or immune system? So we really, at the time, we really didn't know. So we did a single cell analysis. We tried to let the microbes to invade the tumor cells and isolate these tumor cells and did single cells and asked if after the invasion of the bacteria, what happened for the tumor cells? And the pathway analysis showing multiple pathways affected by the invasion of the bacteria. For example, the immune-related NF-κB signaling pathway and TNF signaling pathway, and not like a receptor signaling pathway, suggesting that these cells has been invaded by the bacteria. But these pathways are all upregulated by these bacteria, including the Enterococcus. And the data we show that the Enterococcus has the low ability to promote the metastasis. So we think that these pathways, although they might be involved in promoting the metastasis, this might not be the dominant, the major reason. So another interesting pathway we notice is this fluid shear stress pathway, which is upregulated by Lactobacillus staphylococcus and streptococcus, but they are not. This is not enriched by the Enterococcus invasion. So this is correlated with their ability to trigger the metastasis. So we decide to further analyze whether bacteria play a role in this fluid shear stress process, and we know that when the cancer cells intravasate into the bloodstream, they actually experience the fluid shear stress, and there are some studies suggesting that this fluid shear stress is actually trigger and damage the cancer cells, and they reduce their survival ability. So to test the ability of the bacteria in promoting or helping the cancer cells to resist this fluid shear stress, we actually developed a system according to this paper published in 2017. They designed a in vitro circulating system with a pipeline here and using the peristatic pump, and let the cancer cells to circulate in this like a very small tubing. And that mimic the bloodstream, and you can control the speed to apply different strength of the fluid shear stress. And we found that with different strength of the fluid shear stress, these cancer cells actually the survival ability is actually decreasing, suggesting the fluid shear stress is actually damaging these cells, and they can reducing their survival. And interestingly and amazingly, if we use the bacteria invaded cancer cells and let them circulate in this system, we found that a significant increase of the survival ability after these bacteria invade the cancer cells. And this suggests that after the invasion, the cancer cells become stronger and more robust to resist this mechanical stress from the environment. And why? So why usually we think that maybe bacteria invade the cancer cells, the cell maybe become vulnerable or become damaged, and survival may be reduced. But why these bacteria, they invade the cancer cells, the cancer cell become more robust or stronger? And this experiment actually remind me of my experience in the stem cell biology. So when we do the stem cell biology, we usually when we isolate the cancer, when we isolate the normal stem cell and try to culture them to increase the survival, we usually use a inhibitor called ROCK inhibitor, and to block the contraction-mediated cell death. And this has been published by multiple papers suggesting that the mechanical stress during the isolation of the stem cell can trigger the contraction of the cell. This is mediated by the stress fiber, the actin stress fiber, and this contraction can lead to the cell death. And if you inhibit the ROCK signaling pathway, which is triggered by ROA and ROCK, myosin light chain phosphorylation and cell contraction, if you block this, you can drastically by several fold for their survival. So the phenotype we saw in the in vitro is actually quite similar to the survival enhancement and also the morphology of the cells, suggesting that maybe bacteria also intervening the similar pathway to the cytoskeleton and help the cells to resist the mechanical stress. And we try to test whether this possibility by staining the actin filament in the cancer cell after the bacterial invasion. And interestingly, we found that the Enterococcus invasion did not change this thick actin stress fibers within the cancer cells. But the other strains, Staphylococcus, Lactobacillus, when they invade into the cancer cells, we see a decrease of the actin fiber intensity after the quantification is significantly reduced, and suggesting that this cytoskeleton, actin cytoskeleton system has been reorganized from the thick bundles of the actin stress fiber to the thin fibers of the actin meshwork. And this can help this cancer cells to resist the mechanical stress from the environment. And to further test whether bacteria impinge on this signaling pathway through ROA, we did a threat analysis. And this threat analysis indicating that when the ROA is activated, you will see the energy transfer. When the ROA is inactivated, the threat energy transfer will be reduced. And when we did this threat analysis, we found that these bacterial invasion actually indeed reduce the activity of the ROA GTP, GTPase. And this can block the signaling pathway related to the ROCK and the myosin light chain and prevent the and help to reorganize the actin filament network. Okay, so these studies suggest that bacteria can regulate or modulate the cancer cells cytoskeleton and helps the cancer cell to resist the mechanical stresses from the environment and then help the cancer cell to survive in the distal organ. But the question is that the people still questioning that whether this effect, we saw, whether by antibiotics treatment or the injection or re-administration of the bacteria into the system, maybe this effect is actually mediated by a gut microbiome or the immune system, because when you introduce the bacteria, you can seriously trigger the immune reaction. So to further, test whether immune system is involved in this process or not, this phenotype is dependent on the immune system or not, we try to modulate this signaling pathway by enforced expression of a raw gap protein. Without using the antibiotics or the bacteria. In this way, you don't disturb the gut microbiome, you don't disturb the immune system. And by just modulating this signaling pathway using the raw gap P190, and we overexpress it, we see that the actin stress fiber has been significantly reduced after you express this raw gap. And if you inject this tumor cells to the primary site and let them grow tumors and check the metastasis, we found that primary tumor growth remained similar between these two groups, but the cancer metastasis has been significantly increased by the overexpression of the P90, which inhibits the ROA activity and inhibit the downstream cellular contractility and also the cell death, reduce the cell death. And this suggests that modulating the signaling pathway alone is able to help the cancer cells to resist the mechanical stress from the environment and enhance their ability to survive in the lung tissue. So these data suggest that in the mouse model, these bacteria are actually functional and has a significant, important physiological role in cancer metastasis. And whether this has the phenotype has human relevance, we try to find some human data to try to help us to see whether they are also functional or conserved in the human tissue. So we collected the normal breast tumor, and the lymph node metastasis. We can still detect the microbiota in these tissues, and the breast tissue is higher than the normal breast. And interestingly, the lymph node metastasis contains the microbiota is very similar to the breast tumor. They have very high correlation with the breast tumor instead of the normal breast. And this is consistent with the mouse data that when the cancer cells metastasis to the distal organ, they actually can carry the microbiota with them. So suggesting this is a conserved could be a conserved mechanism. So to summarize, our study actually suggests that besides the cellular components within the ecosystem that can, determine the cancer progression. We found that in the mouse tumor model, the breast tumor model, the tumor is also not sterile. They contain low abundance of the bacteria. And this novel component of the tumor tissue or tumor ecosystem can actually exert their function in a unique way by invading into the cancer cells and exert their role specifically into the whole cell to modulate their cytoskeleton and help them to survive. And this is quite distinct from the proposed model previously by the Stephen Paget 100 years ago, the seeds and soil model, which is basically a passive fitness model that the cancer cells, when they go to distal organ depending on the distal organ's environment, and also distinct from this EMT model showing that the ability of the cancer cell acquire the motility and also the stamina by themselves, intrinsic by themselves, and also through the immune system that NK cells, macrophage, MDSC, or platelet, they can all help or facilitate the cancer cell metastasis. And our data suggests that there are other unknown factors. For example, the bacteria will open a novel, interesting direction in analyzing, what are the driving forces for the tumor metastasis in the clinics. In the future, I think it will be very interesting to further, investigate in the human patient whether this pathway play an important role in the tumor metastasis and also the prognosis. Okay. So with that, I would like to wrap it up and to thank my students, one of my excellent, outstanding postdoc at Kun, and two graduate students. One is Yao Bingqing and Dong Tingting. They collaborate with each other and did quite a huge amount of work in this project. And also, I would like to thank my collaborator from the hospital, from Chen Yongyi, Yao Jia, and Liu Yu. They provide very precious samples for us to analyze the human patient data. And the microbiota data was analyzed by Li Nan from High Performance Computing Center in Westlake University, from Li Nan and Li Hang. And I would also like to thank Westlakers Dang Sheng, Hong Tao, and Tao Liang and Ju Feng for very helpful discussions and suggestions in this project. And thanks for the funding, Westlake University, National Science Foundation, and I will be ready to take questions. Thank you.